Hi, I'm Gbola and Amusa. It's my pleasure to introduce our next guests from Kriya Therapeutics. We have Shankar Ramaswamy, who's co-founder, chairman, and CEO. We have Britt, who's chief manufacturing officer, and we have Eric Hughes, VP of Applied Science. So the format of this session is about 40 minutes for a presentation, then a fireside chat together. And then if you have questions during the session, feel free to ask them in the question box below, and I'll do my best to get to them. So thank you for joining, Shankar. Please begin the presentation before the Q&A.
Oh, great. Thanks, Gbola. So we've got a short presentation prepared to introduce you guys to Kriya Therapeutics, and then look forward to the discussion with Gbola. So to give you a little bit of background, as Gbola mentioned, I'm the co-founder and CEO of the company. At Kriya, we're really focused on building a fully integrated platform in gene therapy to consolidate all of the critical elements to design, develop, manufacture gene therapies at scale. And part of what makes us unique is, one, having the fully integrated capability and building that as part of our mission, but also leading the systematic expansion of gene therapy into increasingly prevalent diseases, thinking beyond rare and monogenic diseases where we've seen gene therapies historically have success. And the company's really built on two distinct technology platforms. One is our focus on rational vector design, which we call SERV. And this is a suite of computational tools that we continue to build in-house, as well as a growing set of libraries of off-the-shelf engineered component elements of the vectors. And we'll have more to share on that in the coming months, but that's not the focus of today's discussion. The other technology platform that we have is our scalable manufacturing platform called Stripe. And this is a combination of the infrastructure in our facility here in North Carolina, where we're seated today, but also many of the next generation processes that we're bringing into the manufacturing process to really drive down the cost of goods exponentially and achieve scale of production in gene therapy. And the way that we've set up the company is with a modular platform that really functions as a magnet to attract high quality gene therapy assets from academia around the world. So thinking beyond a single institution or a single geography, really attracting high quality gene therapies throughout North America, in Europe, in Asia, and plugging these into our integrated technology platform for further vector design with SERV and scalable manufacturing with Stripe. And so we're able to take on programs from institutions that have established proof of concept, or in some cases form partnerships where there's a complementary set of expertise and we can design the vectors and manufacture them ourselves. And this is a vision of what we're looking to build, and we can talk about this more during the fireside chat, but we're really as a company looking to build distinct therapeutic area verticals that are built on this modular technology platform, where we have currently programs in metabolic disease, in ophthalmology and oncology, but growing interest and ongoing discussions in CNS and cardiology, pulmonology, a range of other areas, all of these as distinct therapeutic area verticals that leverage the same common technology platform in SERV and Stripe. And we view this platform as having modularity that allows us to scale, where we're not managing a dozen completely idiosyncratic manufacturing processes, but really building off of a platform manufacturing process at the company. Now, this is a view of the pipeline in more detail. Again, we won't spend much time talking about this today, but we have programs, as I mentioned, in metabolic disease, in ophthalmology, and immuno-oncology. And that includes programs for type 1 diabetes, for uveitis, geographic atrophy, and to address solid tumors, with multiple INDs on track for the second half of 22 into early 23. I expect the pipeline to continue to expand as that's a big part of our strategy as a company in consolidating some of the most promising gene therapies around the world. And to give you some preview of what we'll discuss in today's call, Stripe is our high efficiency manufacturing platform. As everyone is well aware, manufacturing is one of the most significant challenges that's functioning as a bottleneck in the field of gene therapy today, with industry processes at the moment that are inefficient, complex, antiquated, and largely academic, with critical limitations in capacity further compounding those challenges. And at Kriya, we're looking to change the manufacturing paradigm for gene therapies, fundamentally starting not with the product and figuring out how to manufacture it, but really the inverse, starting with the manufacturing process and thinking about what products we can plug into that engine. And so we have a fully integrated approach to process development with scale in mind from the very beginning. Again, with the modular process that we have flexibility to plug in different types of AAV products, and a facility here in North Carolina that's already operational and will have the ability to produce multiple products from the 50-liter scale up to 3,000-liter scale with many innovations to really drive down cost of goods in terms of manufacturing methods. Now, this is an aerial shot of the facility here in RTP. It's a fully integrated capability with our research vector core already online and our pilot production suite at 50 liter already online. We'll have our full GMP production online at 500 liter in the second half of this year. And in this facility, we'll have vial to vial capability with fill finish capabilities, integrated QC and analytical testing, and the ability to go up to 3,000 liter scale with multiple GMP suites running in parallel, all with single use systems. So we're very excited to have this type of facility already online and operational so early in our company's evolution. It's what we view as a distinct competitive advantage in the field. And this is a few photos and images from the facility, but as I mentioned, we have scalable processes and equipment, ability to go and support GLP studies all the way through GMP and even commercial launch with end-to-end manufacturing of drug substance and drug product, full vial to vial capability, and concurrent multi-product GLP and GMP operations that will be online this year with all single-use systems and in-house testing. So we're very fortunate to have this. And with that, Gbola, I'll maybe hand it to you to take us through the discussion.
Right. Thanks for the presentation, Shankar. So, I mean, manufacturing's still tough for AAV-based gene therapy, despite all the capital that's flowed into the space. So what's your perspective on why it's still such a constraint and a big CMC challenge?
Sure.
And CMC is a big challenge.
Yeah, absolutely. So, I'll take a stab at that, and Britt, maybe you can comment further. I think this is still a field that is relatively nascent in its evolution. Just to step back, gene therapies have really only had success in the last few years. And so we're in the early innings of this field in much the same way that, we've seen monoclonal antibodies transform over the last couple of decades. And I think there's a lot of parallels to that situation. So the complexities of manufacturing a gene therapy are, I think, well known to those in the field. It's a complex biologic. There's, multiple nuances to each processing step, challenges in both the upstream and downstream, and critical limitation in both capacity, but also talent in this field. So, I think it makes it an operational and scientific and technical challenge, one that, requires significant investment upfront to overcome, many of these challenges. And Britt, maybe you could comment and also maybe just share a bit about your background and perspectives.
Yeah, happy to, Shankar. So hello, everybody. I'm Britt Petty. I'm the Chief Manufacturing Officer at Kriya. I think you hit it spot on, Shankar. I think, the industry is still relatively immature, and sort of the playbook in gene therapy to date has been to sprint forward sort of unoptimized, non-commercialized sort of processes out of academic and research settings. And then those manufacturing platform processes sort of become the basis of your regulatory filing, and it's very difficult to sort of innovate your way out of those constraints. And so the way we're sort of approaching that is to, begin with the end in mind and develop a platform manufacturing process from the very beginning that our different R&D assets pitch into the Kriya platform strike zone, as opposed to trying to just catch whatever asset comes out of R&D and then just sprint that forward. So take an approach with thinking about manufacturing from the very beginning and manufacturability from the very beginning, and then driving that forward in terms of and getting titers up and improving downstream yields and then scalability.
Got it. And maybe Britt just answered the question, but how do you plan to deploy manufacturing processes across different products and different areas? Isn't that kind of complex, or is that the whole point of how you built the system?
Yeah, I'll take a stab at that, which is, I think, core to our thesis as a company, which is to say that I think a lot of the challenges, at least up front in developing gene therapy product, relate to the technology. And what I mean by that is in the design of the vectors, the manufacture of those vectors, and the analytical methods to characterize those products. And then there are nuances, obviously, with each clinical product and clinical development that are, common to any type of modality in developing a product. So, we really view the investment that's required to make any one of these products truly successful in terms of having a high efficiency manufacturing process, making it at scale and low cost. The investments that are required to achieve that are significant and can be deployed to more than one product. And so given that we are making those investments for one product, we really see some immense opportunity and value to be able to deploy that same, investment of resource, capital, team, and technology to think about what other products we can leverage using that same system. And so that's what the thesis really is of the company, to think beyond single products in that regard. I mean, Britt, maybe you could also talk about modularity and how we think about the manufacturing process.
Yeah, there will be, our process will evolve over time. There will be generations of it, but it will be based on really a common foundation. I mean, we'll gain an understanding with respect to specific genes and capsids, including some of our own internal novel capsids that we're developing. But we'll be in control of the next generation processes and how future genes of interest get incorporated into our manufacturing process, and those analytics that go along with that. So building that platform from the very beginning is still foundational and fundamental to how we're approaching this problem.
Understood. So on your new manufacturing facility, you've obviously made a choice not to go with CDMOs. So talk a little bit more, please, about the rationale for integrating it this early. And maybe also could you talk a little bit about your aspirations on capacity from that facility?
Yeah, Britt, do you want to comment on that?
Yeah, I mean, for us, evaluation of the CDMO marketplace it's very capacity constrained right now. It can be 6 months or longer to get in the door. And we really, to build the manufacturing platform process from the very beginning, it's really foundational and key that we have our own internal capabilities in process development to drive the processes and the process development, have control of that IP, and also really reduce the problem-solving cycle time by being able to do all that work internally with our people on our timelines. So it was really critical for us to have the manufacturing capability and infrastructure. In terms of line of sight on capacity, we're starting up the GMP suites at the 500-liter bioreactor scale, and then building out to a 3,000-liter bioreactor scale later this year. So that'd be sort of a capacity on the facility.
Yeah, and I'll just add to that. I think that there were some features that were pretty unique about our ability to get this up online. One is the capital efficiency. So typically, building out a facility would take call it $100 million and 2.5, 3 years to get truly online. We actually moved in the door to a fully operational facility that was in use for a different modality. So there were some changes to be made, to be sure. But we walked in the door last July with a lot of what we needed on day one in terms of the layout of the facility, fully functional process development labs, a pilot production suite, and the GMP renovation is underway right now. That'll be complete relatively soon. But that was a huge, allowed us to really mobilize a facility like this at a fraction of the cost in terms of capital. So we could do that at basically the beginning of the company. The other thing, as Britt alluded to, there's a lot of innovation in the manufacturing technology that we're really focused on beyond solving for the capacity, which I think is a big focus of the industry and many investors, thinking about innovations in the actual manufacturing technology, in the cell line technology, in the downstream process to really allow scalable purification methods, simplification of processing steps. So those are the unique trade secret know-how and IP that we also think is valuable to the company, irrespective of our pipeline assets.
Just following on that theme of particular innovations that are driving costs down, what else is sort of unique to that facility or the Stride platform that might keep costs low? And are you aspiring in the long term to sort of antibody-like margins or pharma-like margins, anything of that sort?
Yeah, I mean, I think, so to answer the latter question first, I think, that is really the goal for us, is really to get to a point in the field where the cost of production is in line with other modalities. And so, that has been a constraint in the evolution of gene therapy. Why I think gene therapies have historically had the reputation of being, relegated only to this ultra niche category of diseases where we've seen tremendous clinical benefit. I think we see an opportunity, if we can get the cost down and consistency of production up, to be in line with more traditional biologics. I think there's a real opportunity to do that. And moreover, I think there's best practices that can be imported from biologics, given the state of the art in that field as well. So we think it's the right time to try to push towards that direction. That's our ultimate goal. And we expect other companies to also make progress towards that ultimate vision, but we hope to be one of the leaders in driving that change. As it relates to the different levers that really we think are important to pull to bring down cost of goods, I think we look at this as pulling as many levers as possible, just from a technology perspective. So, we're very focused on reducing plasmid requirements, getting to reduced plasmid systems, single transfection, plasmid-free systems. So we think that's an important goal in this field. Being able to improve the per cell productivity and peak cell density, improve yields of the downstream, and achieve scale. So really pulling as many of those levers as possible. And there are proprietary innovations, in each one of those dimensions that we'll obviously look to apply to our own pipeline, but also in the future think about how we can partner with other companies and share our expertise with the rest of the field.
Got it. So, on the slide, it does say manufacturing facility in North Carolina, and I'm noticing that North Carolina seems to have some momentum for AAV manufacturing. So could you talk a little bit about the incentives or the advantages of being in North Carolina? And then as a follow-up, is it part of your recruitment strategy for your manufacturing team? So I'll have a follow-up on recruiting in a second, but please go ahead.
Sure. Britt, do you want to comment on that?
Yeah, North Carolina and the RTP area specifically has really had a tradition of excellence in biologics manufacturing for decades. So it's very attractive to gene therapy companies doing manufacturing and process development from that perspective. And then, the geographic location to all the universities is also very attractive. And, for us to be able to attract talent here, the industry, gene therapy as a modality is growing. It's a very exciting time in this field. And I think, the opportunity to work on this novel modality and being on the cutting edge of what we're trying to do with process development and scalability and the innovations in gene therapy is very attractive to the type of talent that we're trying to attract to the company. And I think finally, it's just thinking about building a culture at the company that is also attractive to talent, a culture of collaboration and teamwork and being able to really solve hard problems and work on the cutting edge of industry.
Mm-hmm. Yeah, and I'll comment on that further. I think, we've grown tremendously over the last year and a half. At the beginning of last year, we were really just 3 founders, myself, Roger, and Fraser. But we're now over 60 full-time employees at the company, about half focused on manufacturing on Britt's team. So Britt, prior to joining us, was the chief technical officer at Avexis and their head of global manufacturing. We've been fortunate to be able to recruit some of the top talent in the field from many of the pioneering gene therapy and biologics companies. I think for us, a big advantage in terms of recruiting assets, but as also talent and people is, one, having the infrastructure to really support that work on day one, essentially. So this is not a build that it will come online at a future point in time. This is really, ready now for operationalization and execution. And I think the second is really a commitment to innovation in manufacturing, that we're not just solving for the supply chain constraints, which are, major problem in this field, but also thinking about new innovations to really evolve the field rapidly towards being like other biologics. So I think that type of mission really appeals to the team that we've recruited, which has been, I think, one of the top teams in the industry, in my opinion.
Got it. And so I think you mentioned 60 people. How much of that is your manufacturing team? So where are you in the process of recruiting for manufacturing specifically?
Yeah, more than half, around half, maybe a little more than half. So 30, I don't know what the latest number is, but in the low 30s are in manufacturing. So, our leadership team in manufacturing, Britt leads the team. We have Mitch Lauer, who was, head of engineering and facilities at Avexis. We have Eric, who joined us from Biogen, Joe Rabinowitz, who joined us from Pfizer, Steve Soltis, who joined us from Avexis, and Harold Alterson, who joined us from Bluebird. So those are the members of our leadership team, really across technical operations, quality, process development, process sciences, and applied sciences. So I think, in each of those verticals have been able to recruit, key talent on the, even the junior scientist level, all the way up to very senior folks at the company.
Got it. And so you mentioned some similarities to biologics, and obviously there are going to be some differences as well. But given where AV is, what lessons can you take from how biologics have been manufactured over the decades?
Yeah, Britt and Eric, do you guys want to comment on that?
Yeah, Eric, why don't you comment on that one?
So, I think in biologics over about the last 20 years, you've seen sort of 100-fold increase in titer levels, at least within the monoclonal antibody spot, right? And I think, as those titers have improved, right, and the, process controls or the controls around the process have improved, the CMC space has matured, the regulatory environment has matured, as far as expectations, right? It's all gone together and sort of maturing the space. So I think, we will see that progression occur, in the AAV gene therapy space going forward. And, you'll see it as the titers improve similar folds, which allows for improved analytics, improved consistency of process, improved control methodologies, and, a more defined regulatory framework for the approval of those molecules.
Got it. And so I wanted to touch base on a question I've been asking each company. The field is changing quite a bit. So I'm just curious what you see as the most important advancements overall within the realm of manufacturing, of course.
Mm-hmm. I think that there's some advances in terms of, I should mention, actually, just to clarify, you mean what are the advances that we think need to be made, or the ones that are happening?
We'll get to the ones that need to be made, but let's start with looking backwards. Ex post, what do you think has been important for you in terms of achieving your goals or for the space overall?
Mm-hmm. Yeah, I'll start, and Britt and Eric, you can add. I think that we're at an interesting inflection point in the field of gene therapy where we are not operating in the dark anymore. I think many of the pioneering companies in the space, the first generation companies, have really laid the foundation for future success in terms of regulatory path, characterizing some of the key bottlenecks and challenges in the field, and then the talent actually now at a point where there's actually a critical mass of talent. It's not an infinite pool of talent, which it's why we've recruited as aggressively as we have. But I think there are people who really know what they're doing in this space, and so I think that has been a recipe that we see as really one for driving significant and exponential and innovation in the next chapter ahead, as we sort of build to the future of gene therapy. So it's really consolidating those key pieces. And as it relates to some of the known levers to pull, I think the ones that I described earlier, now we really have clear line of sight and targets in mind that we want to get to in each of those parameters and many others, where previously I think it was just an open question as to what was achievable. Now there's a clear goal and targets and lines in the sand that we think we can get to that would really move the needle in this field. So it's a little bit of a high level answer, but Britt, do you want to comment further?
Yeah, I really think it's really sort of what Eric was alluding to. It's applying and leveraging technologies that have been deployed in biologics manufacturing for some time now, right? So it's getting to it's cell line engineering to improve transfection efficiencies and cell densities. It's more productive cell lines, better screening, more robust media and feed strategies, allowing you to get to higher cell densities, higher throughput in the manufacturing process. So streamlining the process to get more throughput over a time-based scheme. So, it's really thinking about the technologies that have been utilized in biologics manufacturing for decades, and then trying to mature and professionalize gene therapy manufacturing using some of those same technologies and deploying them here has been really helpful to how we've been approaching manufacturing.
Got it. And with regards to, let's say, the industry growing pains and companies that have hit some production issues, how comfortable are you that what you're doing is sufficiently different that you've lowered the risk of doing the same?
Yeah, Britt, do you want to comment on that?
Yeah, I think and then I'll let Eric add some color. I mean, foundational to our strategy is to robustly characterize, our product quality attributes and our process quality attributes, and doing the right analytics up front so that you have a repeatable and reliable process, and what levers are important to pull in terms of your process performance and reliability and repeatability. And that's really important to do that up front, fully characterize both your process and your product. And I think that's been really helpful for us in our approach of how we're doing and performing analytics. I don't know if Eric, you want to add any color to that?
Yeah, I mean, I think we're being very conscious of the choices we make in the platform, and I think that's going to result in a very well-characterized manufacturing process and a consistent one. So you'll implement modern controls. You're putting a significant focus on the process you're going to deliver at the end, and all the implications of your controls around that, rather than sort of taking what you can get and sprinting assets forward, as Britt previously said. So I think, by being conscious in how you designed your endpoint and you integrate that across the infrastructure you build, the process you design, right, your cell line advancements, et cetera, I think you'll make the right choices and deploy the right strategy as long as those are integrated to avoid some of the pitfalls, from batch variability, low titers, relatively, undeveloped control strategies and identification of CQAs and testing of material. So I think designing your whole sort of end-to-end process with those constraints in mind will help you deliver therapies in the end with fewer problems on the manufacturing side.
Understood. So there's an inbound question. It is, how do you think of SF9 and Baculose system compared to HEC-293 systems?
Yeah, I mean, I think, with the insect cell versus mammalian cell systems, I think there are some, pros and cons. I think, and there are other cell lines that extend beyond HEC-293 and SF9 that, we think are worth considering that, we're also exploring. But Britt and Eric, do you want to take a stab at answering any of that?
Yes, I'll give a stab at it. I guess I would say, some of the things I've seen is sort of, viral, the additional burden of having viral stability on your, SF9 approaches, maintaining sort of your supply chain in an attacked way of how you're going to, service the life cycle of your product among, the various components there. Obviously, right, there's an interest there due to the productivity, and the work that's been done there. But, not being a, hands-on expert in that area, I think there is a reason that a lot of the people in the industry, continue with sort of, more standard methods of AAV production, producer cell lines, et cetera. So I think there's still some work to be done, on sort of the CMC and regulatory path and the SF9 workflow. But I mean, it's obviously of interest, right, and applicability. So, and there are companies using it. So I think not our choice, but not worth keeping an eye on.
And then I wanted to look forward. So ex ante this time, could you talk about where you think the field is going? And I don't mean in a month or 1 year, but maybe 5 to 10 years down the line. What important innovations in AAV manufacturing could help you meet or exceed your goals?
Yeah, I mean, I think that, again, just to harken back to another example that many talk about in this field, I think AAV, the AAV field today resembles in many respects where we were with MABs 25 years ago. And so a lot of the lessons learned from that experience that Britt and other members of the team were a big part of evolving that technology to the mature state that it's in today. Many of those experiences can be brought to bear in this field. And so, I think that's a picture. If you just trace the arc of that, modality and where it is today, I think we would hope AAV in the next, hopefully sooner than the next 20 years, since, this is many of those learnings can be applied here, can evolve to a point where there's consistency of production, scale of production, low cost of production that really does mirror the types of yields and titers and productivity that we're seeing in the MAB field, and or even approaching towards that ultimate goal. So I think if we get to that point, then we can really think about gene therapy as a modality to address almost any type of disease, irrespective of patient prevalence and population size. So that's what we hope is the future of the field. I think it's a big commitment that we're making here at Kriya, but I think others will as well. Yeah, anything further to add, Britt?
No, I think that's well said. I mean, I just say, we're really focused on creating, with our platform, the initial conditions that put us in a position to scale and succeed in the COGS reduction and increase productivity at a pace synchronized with our assets and our programs in preclinical development. So those innovations that are coming, we can incorporate those and they're really going to be the innovations in manufacturing that alleviate the bottlenecks to being able to scale to larger batch sizes and more simple manufacturing processes.
And I think, I think they said it well, but just things will accelerate, right? I mean, Shankar talked about sort of with respect to monoclonal antibodies, but I'd also say it's sort of delivering, rather than CMC and, process being constraints in delivering therapies to the market. You'll see that move. So as the platform becomes well developed, you'll just have to adapt it, which will be a far faster process, and things will move faster through the ecosystem rather than redeveloping everything per asset or per therapy. Right? Acceleration will exist in the CMC space as well.
Got it. And then final question. So Shankar, what do you think is most underappreciated about the Kriya story, whether it's manufacturing related or not?
Yeah. I mean, I think we're still a relatively young company under the radar. So excited to meet more folks as Kriya evolves. But I think I think the evolution of the company, even in the last year, has been pretty significant in every dimension. And what I emphasize in terms of what we're looking to build as a company, it's a fully integrated engine in gene therapy. I think we're big believers in having all of the critical elements to design, develop, manufacture, conduct the right analytical methods to support gene therapy and really move the needle in each of these areas. And so we see value to the current assets in the pipeline. We see this as an engine that allows us to mobilize at scale multiple additional assets and continue to feed this engine with more therapeutics. But we also see value to the platform irrespective of our pipeline assets as it relates to some of the serve technologies, many of the manufacturing technologies that could be the basis for future partnerships that we'll explore. The data that we generate across this platform feeds into many of the computational systems that we're building within the company with our team in California. And so that fully integrated capability is something that we see as obviously a grander vision in gene therapy, but really looking to other modalities, to the types of innovative leading companies that have transformed those other modalities, be it mRNA or monoclonal antibodies or other types of biologics that we can really aim to be that type of company in gene therapy. So that's something that perhaps is underappreciated at this point, but it really is core to our vision in the immediate term, and we're starting to build towards that.
Great. Thank you, Shankar, Britt, and Eric.
Great. Thanks, Gbola.
Awesome. Thanks.
We now conclude this session.